In this talk, I'll be covering the following outline. I will first cover a brief introduction about resting state functional connectivity. Second, I will cover several applications of resting state functional connectivity. Third, I will cover some of the limitations of resting state functional connectivity. And finally, we'll cover some of the current work from my lab that I think are interesting. Obviously, this is very subjective. A typical structural MRI image, which has high spatial resolution, takes a few minutes to acquire. The functional MRI images, on the other hand, has slightly lower spatial resolution, but is collected about every 2 seconds. And the basic idea of bold-based fMRI is that when a subject performs a task, there is increased neuronal firing in specific regions of the brain that leads to increased oxygen consumption, that leads to vasodilation. And this results in an excess of oxygenated red blood cells compared with deoxygenated red blood cells. This leads to an increase in fMRI signal. This is an example of finger tapping task activation, where a subject is presented with two conditions, condition 1 with no finger tapping and condition 2 where there is finger tapping. A number of statistical methods exist to compute voxel-wise measures. A threshold is then used to derive an activation map. And based on the activation map, we can say when a subject performs this task, these brain regions are activated. We decided as a student, my focus was to look at the noise characteristics in the brain, and we studied noise characteristics from 3 different regions. The bottom line is noise characteristics from a voxel outside the brain. As expected, the noise is smaller compared to physiological noise. The white matter noise is significantly less compared with gray matter. For gray matter, we observe the cardiac signal at 1 Hz and its harmonic at 2. We can also see the respiration signal. We can also observe this low frequency signal. And here is a typical time series from the sensory motor cortex during rest. In addition to the respiration signal, we can also observe the low frequency signal. Then what we did was we processed the resting state time series in an identical fashion as we processed task activation data. So what we did was we took the time series data from the sensory motor cortex and correlated with every voxel in the brain. What we found was a substantial overlap between task activation image obtained using bilateral finger tapping and the resting state data, where a time series was chosen from the sensory motor cortex. We found similar results from visual and auditory cortex also. Because Carl Friston had defined functional connectivity as temporal correlation between task-related signal, and we were observing these at rest, we defined it as resting state functional connectivity. Thus, from a single resting state scan, we could identify a number of distinct functional networks, including visual, motor, default mode. In task activation, we would typically require one run, a separate run for each network. Thus, while task activation fMRI has revolutionized our understanding of human brain function, task fMRI has some limitation. In a number of populations, including newborn infants, very old subject and patient population like Alzheimer's, severe TBI, coma, there are issues of compliance. In these cases, resting state fMRI can serve as an alternative for mapping brain function. This is earlier work showing 3 groups of subject and their corresponding resting state network. And the point is, what we can do is assess the different functional network and see differences between visual network, default mode, motor, and executive control, and compare between the different groups. Here, the resting state connectivity pattern in the sensory motor cortex are compared with bilateral finger tapping task in tumor subject. The overlap between the resting state functional connectivity and task activation map are shown to have substantial agreement between the two. In this important study, Helen Zhou and Bill Seeley demonstrated differences between 5 different neurodegenerative diseases, including Alzheimer's and frontotemporal dementia. And the point here is that functional connectivity can be used to differentiate between neurodegenerative diseases and therefore has good specificity and sensitivity. In this work, He-Young and colleagues showed differences in individuals at risk with Alzheimer's. In recent years, due to its translational capability, a number of investigators are using animal models to study connectivity. In this study, Marcus Frakel and colleagues used macaques to investigate functional connectivity with different depths of anesthesia, and they demonstrated the presence of anesthesia modulated functional connectivity. In this study, in collaboration with Young-Ro Kim at MGH, we used rats to investigate connectivity. The left column shows the frequency spectrum, while the right column shows the time series. We show the frequency spectrum during normal breathing. The second row is during exsanguination, where blood is withdrawn, showing an increase in fluctuations in the low frequency range. And the bottom, after the blood has been replaced back into the rat. And again, these reversible change that we observe suggest that these fluctuations are physiological in nature. In this study, with Chris Pavella, we used rat to investigate functional connectivity. The leftmost column shows task activation for sensory motor regions. The second and third column show the corresponding functional regions, showing as to how functional connectivity covers more of the functionally related regions. Another application of resting state signal is the ability to calibrate the task activation response. The fMRI signal observed is a combination of neuronal firing convolved with the underlying hemodynamic response function. In fMRI, we typically assume the response is due to neuronal change. One method to account for variation in the underlying hemodynamic response has been to estimate it using the hypercapnic response. Hypercapnic response, either using CO2 inhalation or breath holding, gives a reliable estimation of hemodynamic response function. We can then scale the fMRI response by dividing the bold signal during task with bold change during hypercapnic stimulus. Thus, we wanted to investigate if we can substitute resting state instead of a hypercapnic stimulus. We show time series from breath holding and 5% CO2 inhalation, and this figure shows a scatter plot between the two, showing strong correlation, suggesting that one can substitute one for the other. A voxel-wise map of changes during task is shown for finger tapping and cognitive task. In addition, we show the standard deviation during for those voxels. A substantial overlap between the two, between the task change and the standard deviation during rest is shown. Here we have demonstrated calibrate response of task response using breath holding on the middle row and resting state calibration on the bottom row. Again, the point I would like to make is that there are compliance issues in hypercapnic stimulus. Some subject cannot hold their breath, while some subject may get claustrophobic when putting a gas mask for CO2 inhalation. More importantly, resting state data has other application. Even if one does not believe in resting state functional connectivity, we can still use it to calibrate the task response. So it is one thing to have one's paper rejected or grant rejected. It is a whole different problem when the editor-in-chief of the primary journal in your field, criticizes your work. Paul Fletcher, the main complaint was, since everything we know about neuroscience is from task response, why bother? The second point was, what does resting state signal mean? The third point was, how do we correct for artifact when we do not know what the ground truth is? And finally, the complaint was that since most studies were done on 20 to 30 subjects, by the way, the same as is done for task activation study, they thought it may not hold true for 500 or 1,000 subjects. So since you cannot really argue with a reviewer, let alone the editor-in-chief, we decided to investigate the reliability and reproducibility of resting state measures. We asked every research group that had published resting state data in 2009 and before, and got a lot of data. The scanner were different: GE, Siemens, Philip, one variant, different field strength 1.5, 3T, 4T, 7T, different imaging parameters, different trace, different time of day, and analyze the data in an identical fashion. And we found highly reproducible resting state network. Again, this is from more than 1,000 subjects. Because we had so many subjects, we could look at effect due to gender and age. On the left, differences between male and female are shown. On the right is shown effect on connectivity due to age. Yellow shows region where connectivity is reduced with age, while brown color shows region where correlation increases with age. Again, all this data is publicly available on NITRIC. Here I'll briefly discuss some of the work done to differentiate between neuronal basis versus vascular effect. Again, the fMRI signal can be thought of as a convolution between neural activity and physiological factors. Here, in an experiment, we used hypercapnic stimulus to study the effect of increased blood flow on functional connectivity. First, task activation was performed using bilateral finger tapping. Then resting state functional connectivity was obtained. Then during 5% CO2 was performed. And what we can see is a significant reduction in functional connectivity. 5 minutes after resumption of room air breathing, a resting state functional connectivity was again observed. the fact that we could reversibly change the functional connectivity suggests that the functional connectivity has a biophysical mechanism. In this study, Vesak-Evanimi used thiopental to show that there was a significant increase in functional connectivity in the visual cortex. In another important study, Scott Peltier demonstrated that the functional connectivity was modulated and decreased as the depth of anesthesia increased. Again, the point is that with different anesthesia, there is a change in connectivity. So in the next 15 minutes or so, I will talk about some of the areas of research that I think are interesting. Again, there has been some debate as to what is task activation map and what is resting state connectivity map. Some have suggested that resting state functional connectivity map is a superset of task activation. Others have suggested while task activation is used for functional localization, functional connectivity is used for functional integration. To study the link between task and rest, we used a brain mapping database. Using a large number of studies, it stores the location of all active region. For any location, Neurosynth identifies all other regions that were also identified as active when the specific region was chosen. So for this study, 140 commonly used regions of interest was identified along with the corresponding co-activation regions. This shows how the data was extracted both for co-activation and for resting state. The same 140 regions was used. What we show here is the correlation between co-activation and resting state correlation matrix. A significant correlation between co-activation matrix was obtained. What we wanted to see if there was some difference between co-activation network and resting state correlation matrix. In this case, we use modularity to study the differences for resting state networks, for co-activation, and a random noise network showing how there are differences between them. Further, what we can show is that the thalamus in activation, co-activation was more active compared to the resting state. So what we found is that the co-activation network has higher global efficiency, lower mean clustering coefficient, lower modularity, and high degree in thalamus, while resting state network has lower global efficiency, higher mean clustering coefficient, higher modularity, and high degree in left temporal lobe. There has been a lot of work in EEG, MEG, where the frequency spectra, due to its temporal resolution, is much wider. Buzaki and colleague have shown that neuronal fluctuation obtained has distributed across a wide range of frequencies. With multiband feature, where we can collect data at a much faster rate, we wanted to see if we looked at frequency higher than 0.1 hertz, would we be able to see any other frequency or spatial pattern. Here we analyze the data according to different frequency band and obtained significant interhemispheric connectivity for all the resting state network. Nonetheless, what we see is that the dominant frequency is still in the lower frequency range, but which is basically below 0.1 hertz. Although higher frequency, there is some spatial pattern, the dominant is in the lower frequency range. This is showing how we can subdivide these frequencies in cases in schizophrenia compared to healthy control, and then show differences between first episode and chronic patients. In this, because Dr. Katie Chang will be talking about functional dynamic connectivity, so I'm just going to cover one small aspect of dynamic connectivity that I think is interesting. And so while others have investigated the role of heart rate and respiration, we have tested the hypothesis if dynamic connectivity is modulated by other brain regions. So what we have tried to address is, are modulatory interaction present in resting state signal, whether modulatory interaction exists between default mode network and task positive network, and whether modulatory interactions are modulated by task demand. And what we show is that we use the DMN to PCC and MPFC and demonstrate that task positive network influence connection between PCC and MPFC. While it is known that task positive network influences DMN, we show that connection between PCC and MPFC are influenced by task positive network. Similarly, we show that salience network influences connection between default mode network and task positive network. Similarly, between eyes open and eyes closed, we show that the thalamus is involved. So using the information that during rest there is so much interaction between regions and network, we tested the idea that there are more widespread regions activated by task that are not detected by traditional method like GLM. So for connectivity model, we have used seed-based correlation, and this is used for whole brain. For task activation, the reference is the task reference. In the PPI, we have the two effects and an interaction between them. And we want to see the interaction between these. And we used data from Human Connectome project. We used one additional stop signal, which is an event related from the UCLA publicly available database. And what we observe is activation very similar to study done by Diana Barch and colleagues at WashU, basically showing the same activation by doing GLM analysis. But then we used PPI analysis, and what we show is ROI-wise activation in 4 tasks that later showed significant psychophysiological interaction. Red and blue regions indicate increased and decreased activation during task condition compared with respective control condition, again suggesting a much more widespread interaction even during task activation. And so this figure for the different tasks shows the relationship between task-modulated connectivity and regional activation level. The upper two rows show circular plots of increased um, um, represented in red and decreased blue connectivity for different tasks. The regions along the circle were ordered based on their activation in each task and were colored based on their significance level. The bottom row illustrates the network layout of the first giant component using Evan-Hughes algorithm. So far, with resting state connectivity analysis, we have challenged the assumption that that the resting state is noise, right? So in the last few minutes, I want to look at the assumptions that we have made ourselves in resting state analysis. So the assumptions, one assumption we have made is that most of the information is in gray matter, and white matter and CSF are noise dominated. However, white matter takes up more than 50% of brain volume. There has been study by Ding and colleague at Vanderbilt where they showed a correlation between task load and temporal correlation with white matter and gray matter region. And Michael Peer, using white matter, demonstrated clusters similar to clusters obtained using ICA. So we wanted to focus on corpus callosum, the largest white matter structure. We used data from the Human Connectome project. We used 119 data which were collected 2 times and used k-means to determine their clusters. A number of white matter clusters were observed. These were found to be highly reproducible for both the 2 runs that we analyzed. Further, we wanted to see whether the white matter networks that we saw in the corpus callosum, whether there was some overlap with the DTI data. And what we show here is the structural and functional connectivity between human corpus callosum and white matter functional network. The top row demonstrate each white matter functional network demonstrates specific correlation in structural and intrinsic functional connectivity with distinct areas of corpus callosum. The bottom row shows considerably overlap between structural and functional map results as demonstrated by the connectivity profile in red. While they are not exact, significant overlap exists between them. The current study demonstrates that corpus callosum connecting different white matter functional network has functional segmentation and spatial distribution pattern. There is a lot of work that still needs to be done to understand the mechanism, either by using physiological perturbation, using clinical application, or using multimodal techniques. Finally, I would like to thank every single of my student and colleagues who have helped me over the years. Special thanks to many investigators who have shared with me magnet time, free data, and many other resources and support. Thank you so much. I really appreciate this opportunity.